Hello, my name is Craig Kundrot. I'm the director of the Biological and Physical Sciences Division in NASA's Science Mission Directorate. Thank you for this opportunity to introduce you at a high level to NASA's Space Biology program. In the division, we use spaceflight environments to study biological and physical systems. By examining phenomena under extreme conditions, like microgravity and exposure to deep space radiation, we can understand how systems function in ways that are not accessible to us as experimentalists on the ground. The resulting understanding can contribute to significant scientific and technological advancements that both enable space exploration and benefit life on Earth. So this gives rise to a two-pronged mission: pioneer scientific discovery and enable exploration. Our scientific discovery efforts are guided by our Decadal Survey and its midterm assessment. The Decadal Survey was produced in 2011 by our National Academies of Science, Engineering, and Medicine. We use this as a guide to proactively seek out new ways to expand fundamental scientific knowledge. As pioneers, we're keen to share this information and know-how, so we seek to provide expertise and support to others seeking to utilize space. The second prong of the mission, enabling exploration, involves anticipating and investigating critical areas for scientific knowledge and technology development for NASA's human spaceflight exploration. So in our world, that can include human physiology, plant growth, and maintenance of the microbial environment on a spacecraft. We deliver results from this research to other NASA organizations that are actually working on the exploration missions. More details about the space biology program can be found in the science plan depicted here on the right and with the URL listed on the bottom. The overall vision is to create new knowledge of how the space environment affects biological systems, and in particular, those biological systems that are relevant to human exploration of space. So that means the human crew, plants that may be along on the mission, and microbes that will be associated with the crew and the built environment of the spacecraft and the plants. We build links between space biology and human research at NASA, so our space biology findings can translate to applications. We essentially go from the mechanistic level of DNA and RNA to applications in flight. A major part of the strategy is to make maximum use of each experiment by depositing the results in databases, either the Life Science Data Archive or GeneLab, GeneLab being our premier database and tool set for omics data. For decades, it's been a central tenet of our program to leverage international partnerships and collaborations in the execution of our work. And lastly, we're very keen to train and inspire the next generation of space biologists. Space biology has many components to it. At the whole organism level, we study plants, animals, and microbes. We also look at simpler systems like cell culture and molecular studies. We also do longitudinal studies in developmental, reproductive, and evolutionary systems. Overall, the flow of objectives is typically to first, for a given system of interest, see how that biological system responds to the spaceflight environment. Those first steps are often just phenomenological. But the second step then is to tease out the underlying mechanisms that prompt that response and develop physiological models for those systems. To do these experiments, we often have to develop cutting-edge technologies to facilitate the spaceflight research. Once the research is conducted, as I mentioned before, we use open science with the GeneLab data system and the Life Science Data Archive to maximize reuse of the data and comparison across experiments and organisms. We use this to provide mechanistic understanding to support human health in space, but also support the transfer of knowledge and technology back to Earth to benefit life on Earth. And at the bottom of this slide, you can see the two URLs for GeneLab and LSDA. To do this work, we use a wide variety of platforms. Our workhorse for spaceflight is the International Space Station. But we also use CubeSats and, in collaboration with our Russian partners, free flyers like the Bion. On the upper right of this figure, you see the future lunar platforms that we're keen to use, the Lunar Gateway and Lunar Orbit, and lunar surface opportunities. Short of those, there's a wide variety of analogs that we use back on Earth, either on the ground or parabolic flight, sounding rockets, suborbital flight, and the like. So we use a wide range of platforms depending on what's appropriate for the research, and we very often use a stepping stone approach where we will begin with Earth-based analogs and eventually move up to orbital flight. As I mentioned before, international partnerships and collaborations have been a linchpin of our implementation strategy for several decades. Since 1990, we've had the International Space Life Sciences Working Group convening together twice a year. This involves the Italian, French, Canadian, German, European, Japanese, and Russian space agencies. As a separate venue for collaboration, we've had the US-Russian Joint Working Group that's met since the early 70s in support of the Apollo-Soyuz test project. One of the more notable aspects of collaboration there is our participation in their Photon and Bion free flyer biosatellite programs. And we are preparing right now for the next Bion flight. So I've pointed to some ways that you can learn about the content of the space biology program, but here are a couple of ways that you could participate directly in the space biology program. First is GeneLab. The URL is listed here at the upper left. GeneLab consists of almost 300 studies with omics data from a wide variety of organisms, mostly from spaceflight, but many from ground or other venues like parabolic flight. In addition to providing the data, GeneLab has developed a lot of tools to analyze the data and help standardization of the analysis approach. In support of that, analysis working groups have been stood up. There are over 120 scientists from multiple space agencies and international institutions that are participating in, roughly speaking, monthly meetings to analyze data in the GeneLab repository. And they've already published a number of papers. And they're very happy to take on new members. So involvement in GeneLab is one direct way to learn about some of the spaceflight findings in the program and to actually participate in the research. Another way to get involved or to learn more about the program is our Decadal Survey. Decadal surveys in the US are instrumental in defining NASA's research agenda for the next decade, and they also lay the foundation for the decades that follow. They're produced by our National Academies of Science, Engineering, and Medicine, and the URL for our current Decadal Survey, which is about to begin, is listed here. This effort should be starting late this year or early next, and a key feature of these Decadal surveys is they are driven by the research community, and not just the US community, but the international community. The primary way the community is providing input is through white papers, and our American Society for Gravitational and Space Research is conducting a series of meetings and providing a lot of resources for the community to learn about how to write these white papers, and equally importantly, maybe more importantly, actually providing a venue to come together and form teams to develop white papers of mutual interest. And the URL for the society and the Decadal Survey effort is listed here. And your involvement is welcome. You could be involved just to take in information or be an active participant in shaping ideas for future research. So to sum up, space biology studies a broad range of organisms, from the molecular level to, in the case of microbiology, the ecosystem level. We utilize a broad range of experimental platforms to do this work, ranging from those on the ground to those in spaceflight. We have a rich history of international partnerships that thrive on mutual benefit, and we welcome the opportunity to explore collaborations with Australian researchers and platform providers. 3 ways to engage space biology are to use the GeneLab database and toolset, and to join the analysis working group part of GeneLab. Second, follow the Decadal Survey process and consider providing input. And lastly, I'll give you 2 contact people that you can follow up with that would be delighted to hear from you. Sharmila Bhattacharya is our program scientist for space biology, and Nikki Rail is our program manager. So with that, I'd like to thank you for your attention and wish you the best. Thank you.